back. This session is on chromosomal theory of inheritance and linkage. And the learning goals are 1. Explain 3 major similarities between chromosomal behavior and Mendelian factors. 2. Analyze the experimental evidence for the chromosomal theory of inheritance. 3. Enumerate the advantages of using Drosophila as a model organism. 4. Describe genetic linkage in body color, eye color and wing size genes in Drosophila melanogaster. 5. Differentiate recombination frequency in tightly and loosely linked genes. And 6. Determine the basis for construction of the first genetic map. When we think about the science of genetics, it dates back to 1865 when Gregor Mendel first proposed with his fundamental laws of inheritance in pea plant, much before the discovery of chromosomes and genes were made. But with the advancement in microscopy, many scientists started looking into the internal cellular structures and their functions. A German anatomist named Walter Flemming used aniline dyes in order to stain the cells of salamander embryos. And Flemming was the first scientist to have visualized thread-like material inside the nucleus during mitosis. He called these colored thread-like material as chromatin. And chromatin literally means colored substance. So much of the understanding that we have today about the movement of chromosomes during mitosis originates from the work of Flemming, which was published in the year 1882. However, Flemming failed to understand the role of chromosomes in heredity. Another German embryologist named Sutton-Boveri took Flemming's finding to the next level. Boveri demonstrated that both sperm and egg must contribute equal number of chromosomes for the proper development of embryo in sea urchins. He also recognized that Mendelian factors are carried on the chromosome. Around the same time, in the year 1902 and 1903, an American graduate named Walter Sutton observed the separation of maternal and paternal chromosomal pairs into the daughter cells during meiosis in grasshopper. Now, independently, the observations of Boveri and Sutton led to the development of chromosomal theory of inheritance, which is also known as Boveri-Sutton chromosomal theory. Chromosomal theory of inheritance is a fundamental unifying theory of genetics which states that chromosomes are linear structures with genes located at specific sites and they are passed from one generation to the next generation. In fact, Sutton and Boveri noted striking similarities between the chromosomal behavior during meiosis to that of Mendelian factors during inheritance. One such similarity is chromosomes, like Mendelian factors, also occur in pair in an organism. So, if we take an example of the stem height, in pea plants, stem height is controlled by a pair of factors. Now, here we have a heterozygous pair of factors, capital T and small t for stem height. Likewise, in an organism, Sutton and Boveri propose that chromosomes also exist in a pair. A pair of such chromosomes having same size and having centromere at the same position are known as homologous chromosomes. Now, on each member of such homologous chromosomes, we can be also expecting Mendelian factors, or we can be expecting the genes to be carried on specific location. This location of the gene on a chromosome is referred to as a locus. So here you can be, in this figure, you can be observing with the two factors, capital T and small t, carried on each member of the homologous chromosome. So in this way, Sutton and Boveri proposed with the similarity between the existence of a chromosomal pair to that of the existence of a pair of Mendelian factors. The second similarity is the members of a homologous pair segregate during meiosis and each gamete would then be receiving only one member of the pair. And this is very similar to that of Mendelian law of segregation, where a pair of factors would be segregating during gamete formation. Now, the pair of factors capital T and small t in the parent would be segregating at the time of gamete formation, and each gamete will receive only one of the two factors. So, we have a gamete having just capital T and another gamete having the small t. Similarly, even the pair of homologous chromosomes which are present in an organism at the time of gamete formation, especially during meiosis, they are going to be segregating or separating from each other. And this separation or segregation of the homologous chromosomal pair occurs during anaphase 1 of meiosis 1. And as a result of which, each of the daughter cell or each of the gamete would only be receiving one member of the homologous pair. Okay, and here in this case, so this gamete has the chromosome, one of the member of the homologous chromosomes having capital T factor, and the other gamete got or received the other member of the homologous chromosome having small t factor. The third similarity between chromosomal behavior during meiosis to that of Mendelian factors is that one member of a chromosome pair can independently assort with any member of another chromosome pair during gamete formation. And this is very similar to that of Mendelian law of independent assortment. Now, from the Mendelian context, what we know is when we have two pairs of factors in a plant, at that time, one pair of factor can independently be segregating from each other, and one member of the pair of factors can independently be assorting with any member of the other pair of factors. That is to say, capital T can independently be assorting with capital Y, or capital T can independently be assorting with small y. In the same way, small t can independently be assorting with capital Y, or small t can independently be assorting with small y. And in the same way, we can speak in terms of capital Y independently assorting with capital T or small t. Likewise, small y independently assorting with small capital T as well as small t. And in that way, we are expecting 4 different types of gametes in equal proportion, and this is what we know as law of independent assortment. Now, similarly, when we consider 2 pairs of chromosomes in a cell, for example, here the 2 pairs of chromosomes are denoted by 2 different lengths of the chromosome. So, one of the pairs have the longer length, so we have a pair of longer chromosomes, and the other pair has shorter length, so we have a pair of shorter chromosomes. And what we also want to be noting here is I have assigned with the particular factors or genes on the chromosome. So the red long chromosome and the its homologue, that is the black long chromosome, has the T factors. Okay, so these are the chromosomes having the let us say the stem height gene. So red has capital T, black has small t allele. Whereas the 2 shorter chromosomes are denoting with the 2 alleles or factors of seed color. So one has, green has capital Y and blue has small y. Okay, so now with this in mind, now let us say that when we have such pairs of chromosome during meiosis, particularly during metaphase 1 of meiosis 1, when the homologous chromosomes are going to be aligning at the equatorial plate, at the metaphasic plate of the cell, we can be expecting 2 different kinds of patterns or 2 different kinds of alignments. So this is one of the possibility, and this is the other possibility of the alignment of homologous chromosomes at the metaphasic plate. So now let's first take this type of alignment. Now in this type of alignment, what you observe here is I have placed a red long chromosome and green short chromosome to one side of, or to one pole of the cell, whereas I have placed black long chromosome and blue short chromosome to the other opposite pole of the cell. And as a result of which, during the time of anaphase 1, when we have the separation of these homologous chromosomes, now what is going to be happening is, because of such alignment, a long red chromosome would always be assorting with or combining with, moving together with green short chromosome. Okay, and as a result of which, we are to be expecting a certain proportion of gametes having the combination of red long and green short chromosomes. And as we know, red and green chromosomes are going to be carrying their respective alleles or genes. Okay, so the alleles on red and green are capital T and capital Y respectively. And as a result, we are going to be having the gametes which are going to be having the factors which are capital T and capital Y combination. Likewise, when the long black and long blue are going to be pulled to the opposite pole, so the other daughter cells are going to be having black and blue combination of chromosomes, or black and blue combinations of let us say the factors. And these gametes are going to be having the combination which is small t and small y combination of gametes. Okay, so this is one possibility of the alignment of homologous chromosomes during the time of metaphase 1. The second possible alignment of homologous chromosomes during metaphase 1 can also be of this type, where I have swapped the, if you observe carefully, you can see that I have swapped the two smaller chromosomes. So now, instead of green aligning towards the same pole with red chromosome, we have blue short chromosome aligning to the same pole with that of the red long chromosome. And because of such kind of alignment, now during the time of anaphase 1, red long and blue short are going to be pulled towards one pole or towards one daughter cell, and black long and green short are going to be pulled towards the opposite daughter cell or to the opposite pole. And because of which, such kind of cells are going to be producing gametes which are going to be having the combination of red and blue and black and green combination, where red and blue is the combination of the factors capital T and small y type of gametes, and black and green combination of the gametes are going to be having the factor combination which is small t capital Y combination. So in this way, we can see that we have 4 different types of chromosomal combinations. Right? And these 4 different types of chromosomal combinations are all possible because of the independent assortment of one member of the chromosome pair with that of another member of another pair of chromosome. And this would be resulting in 25% of capital T capital Y type of chromosomal gamete combination, small t small y type of 25% of small t small y type of chromosomal gamete combination, and then 25% of capital T small y type of gamete combination, chromosomal combination, and another 25% of small t capital Y type of gamete chromosomal combination. And this is in a way suggesting with the kind of demonstrating with the kind of similarity that is there with Mendelian law of independent assortment. Thus, chromosomal theory of inheritance demonstrates that genes resides on chromosomes, and the behavior of the chromosome during meiosis underlie the pattern of Mendelian inheritance. Despite such strong correlations in the behavior of chromosome during meiosis and the behavior of Mendelian factors during inheritance, chromosomal theory of inheritance was not widely accepted in the initial years. And this was because it lacked experimental evidences. And many scientists were skeptical about the existence of far more number of traits or genes in an individual to that of the total number of chromosomes they had. Scientists thus needed an experimental system in which the inheritance of genes or traits could be directly linked to the behavior of chromosomes. Experimental evidence for the chromosomal theory of inheritance came from the contribution of yet another American geneticist named Thomas Hunt Morgan. Morgan and his colleagues worked on the genetics of fruit flies. The scientific name of fruit fly is Drosophila melanogaster. So while working on the genetics of Drosophila, Morgan proved that genes are located on the chromosomes in the year 1910. And before we go into the details of Morgan's genetic studies, let's first try to understand why Drosophila becomes such a good model organism. The reason are these flies have a very short life cycle. So in just few days, in around 10 to 12 days, they can be easily becoming into an adult. And they also have the capacity to produce large number of offsprings. In fact, each female can be producing up to 100 eggs per day. And such flies have a very simple genetic makeup. These flies are just constituted by 4 pairs of chromosomes, which becomes very easy and convenient for genetic studies. And having said that, they also show many inheritable variations in the population. And these flies are sexually dimorphic. So we have male and female flies, and male and female flies can easily be differentiated by looking at the physical appearance. So male flies are relatively smaller in size, and male flies also have a dark pigmentation at the posterior end of their abdomen, which is not seen in females. And another very important feature is that they are very cost effective. So these flies are very easy to maintain, and since they are very small in size, we can be raising hundreds of these flies in a small bottle or in a small vial. And not only that, they can be easily living on, or they can be very cheap in terms of their food requirement, because they can live on simple cornmeal agar mixture. So for these reasons, Drosophila becomes the most extensively used and one of the most well understood of the model organisms. Let us now figure out how Morgan provided experimental evidence for the chromosomal theory of inheritance. Morgan knew that there are 4 pairs of chromosomes in Drosophila. Of the 4 pairs, 3 of the pairs are autosomal chromosomes. Okay, these are all the chromosomes which are non-sex chromosomes, and 1 pair becomes the sex chromosomes. Now, in Drosophila, females usually have 2 X chromosomes as their sex chromosomal pair, and males have 1 X and 1 Y as their sex chromosome pair. Now, while doing his genetic studies on Drosophila, Morgan realized that most of the flies had red eye coloration. And because of which, because red eye color is the common eye color in the population, such eye color is referred to as wild type eye color. So wild type refers to the normal phenotype or the common phenotype in a population. And in his population, he did find some of his flies having white-eyed coloration as well. And since white eye coloration is a deviation from the normal red coloration, these eye color are referred to as mutant eye color. Okay, so mutant refers to the deviated or the different eye color to that of the wild type or normal eye color. Now, what we need to keep in mind here is we have a slightly different notation to be used for Drosophila genetics to that of the Mendelian genetics. Now, when we have to denote a wild type phenotype, for example, let's say we have to denote red eye. So, red eye would be denoted by a letter having a plus superscript. So, a letter with a plus superscript would be denoting wild type phenotype. Okay, and a letter without the plus superscript would be a mutant phenotype. So now in the Drosophila genetics, commonly what we tend to be using as the letters for a particular phenotype is, we are going to be using the first letters of the mutant phenotype. Now, out of the two eye colors, red and white eye color, we are going to be using the first letter of the mutant phenotype. So, W. Okay. And if we just have a W written there, it means it is a mutant eye color. It refers to white eye coloration. Whereas if we have a W with plus superscript, that has to be referring to the wild type eye color or red eye color. And if we have the letter written in small or the lowercase letter, then it has to be indicating that the trait or the phenotype is recessive. Whereas if we have a letter which is written in capital form, then we have to know that the phenotype or the trait is dominant. Now here, white eye is recessive to red eye because it is denoted with a small w. Okay, so a small w refers to white eye color, and this is a mutant phenotype, and a small w with plus superscript denotes a wild type red eye color. And we understand that red eye color is the wild type eye color, and this is the dominant eye color over white eye color. Here is an illustration of the first experimental cross conducted by Morgan in Drosophila to study with eye color inheritance. In his first experimental cross, what Morgan did was he crossed red-eyed female to white-eyed males. And by crossing such parents in the F1 generation, he observed that all of the F1 flies had red eye coloration. Now on doing F1 selfing, that is by crossing F1 red-eyed female to another F1 red-eyed male, he was able to obtain F2 generation. Now in the F2 generation, what Morgan observed was 3/4 of the F2 flies had red eye coloration and 1/4 of the F2 flies had white eye coloration. Now this result, F2 phenotypic result, was very consistent with that of Mendelian monohybrid ratio. It was 3:1. However, Morgan was surprised to find a strange result in his F2 generation. He found that the inheritance of red and white eye coloration was unequal in F2 females and F2 males. Of the total F2 female flies, all of the F2 females had red eye coloration, whereas only half of the F2 males had red eye coloration and the other half of the F2 males had white eye coloration. So now Morgan have to determine why only half of the F2 males had white eye coloration. To determine why there was an unequal inheritance of red and white eye color in Drosophila, Morgan and his colleagues conducted many other genetic experimental crosses in Drosophila, of which one such cross that gave them the strongest base to explain where the inheritance of eye color was a cross between white-eyed female and a red-eyed male. So when a cross was made between white-eyed female and a red-eyed male, what they noticed was, in the F1 generation, all of the females had red eye coloration and all of the males had white eye coloration. So this result, where all females had red eye coloration and all males had white eye coloration, led to the confirmation or to the conclusion that eye color gene was inherited in the same pattern to that of the inheritance of X chromosome in the male and female flies. So the way in which X chromosome is inherited in the male and female flies was equivalent to the way in which eye color gene was inherited in the male and female flies. And this is how Morgan concluded that the gene for eye color was located on the X chromosome. To get an idea about Morgan's conclusion that the inheritance of eye color gene and the inheritance of X chromosome in the male and female flies follow a similar pattern. Let's try to illustrate the cross in this way, where we are going to be showing the chromosomes for the respective flies, as well as we are going to be locating or placing the eye color gene on X chromosomes. So we know that eye color gene can be W or W+. So W is for white eye color allele and W+ is for the red eye color or the wild type eye color allele. And another thing that we know is that the females in Drosophila have 2 X chromosomes, whereas males have 1 X and 1 Y chromosome. Now, out of the 2 X chromosomes in the female, we realize that 1 X chromosome is always contributed by the female gamete or the female parent, and the other X chromosome is always contributed by the male gamete or the male parent. Okay, so even though we have 2 X chromosomes in the female, each of the X chromosome is contributed by the male and female parent. Now in the males, we have 1 X and 1 Y chromosome. Now in the males, the X chromosome is always to be contributed by the female gamete or the female parent. So please take note of this. For males, they have only 1 X chromosome, and this only 1 X chromosome is always to be contributed by the female gamete or the female parent, and the other Y chromosome, which is their other sex chromosome, this is contributed by the male parent. Okay, so for males, X are always to be contributed from the mother, alright, and from the father they receive Y sperm. With this knowledge, now let's try to look into Morgan's cross once again. So we said that Morgan crossed red-eyed female with white-eyed male. So red-eyed female had homozygous red combination, that is W+, W+ on both of the X's, and white-eyed male, so they have only 1 X chromosome, and these parents had on their X or only X chromosome, they had the recessive W allele, which is for white eye. Now we have to trace, or we have to track with the pattern of X chromosome that is inherited into the male and female flies, as well as the pattern of eye color gene or the eye color allele that is inherited from the male and female parent. So in the F1, we do see that all of the males and female flies have red eye coloration. The reason is because when we plot, when we have the use of Punnett square and then try to have all the possible, predict all the possible combinations of genotypes, so we find that all of the females are heterozygous. So even though they are receiving one of the X containing this white allele from the male parent, even though one of their X chromosome is having the white allele, but then they are not expressing the white eye color because the other X chromosome contributed by the female parent has W+ allele. And because of this W+ allele, which is present on one of their X chromosomes, so the expression would be always of red eye coloration because red is dominant over white. Now the males are always to be expressing red eye color in the F1 generation because the only X chromosome that they are going to be receiving comes from female parent. And here, since female parents are always to be contributing the same type of egg, having W+ allele, so because of which males are also having red eye coloration. Okay, so this is how the males are, F1 males are red because the P generation or their mother had X chromosomes having this red allele. Now in the F2 generation, we see a slight difference because here in the F2 generation, we see that mother has also one of her X chromosome carrying small w. So this mother is F1 mother is heterozygous. And because she is heterozygous, there is every likelihood, there is 50% chance that the females and the males are going to be receiving this X chromosome having the white allele or the recessive W allele. And as a result of which, males that received this egg having the X chromosome with W allele, that is the white eye allele, and since they have only one X chromosome, so these males had white eye phenotype. Whereas these males, half of the males or in the F2 generation had red eye color because 50% of the mother's X chromosome carried W plus allele. And as a result of which, 50% of the males had red eye coloration in F2 generation, whereas half of the 50% of the males had white eye coloration. So this 50% of or half of the males that were only showing with white eye coloration is because of, again, the X chromosome in the mother that carried with the W or white allele. Okay, so this is how we can track with the similar pattern in the inheritance of X chromosome and the inheritance of eye color. So if you receive this kind of X chromosome from the mother, you are to be also receiving the white allele. If you receive this type of X chromosome from the mother, you are also to be receiving that type of allele from the mother. Okay. Likewise, we can be also looking into one of the other experiments that was conducted by Morgan, and basing on which they came up with such kind of a conclusion. So here they used a homozygous white-eyed female and crossed it with a red-eyed male. So now in this female, both of the X chromosome has small w allele, and because of which this female have white eye phenotype. So female can only be expressing white eye phenotype when they have W allele, that is the recessive white eye allele on both of its X chromosomes. Okay, whereas if we have one white eye allele on the X chromosome in the males, they are to be automatically expressing with white phenotype. But in females, even though they might be having one of the white type allele in one of their X chromosomes, but then the phenotype would not be expressed as white eye because white eye is recessive to red eye. Okay, so here it has to be always homozygous, and as a result of which female parent this time would be always contributing the same type of egg having W allele or white eye allele. And as a result of which, even though the females are to be receiving 50% of the X chromosome in the females are to be contributed from the mother, but they receive also the other. So they have a compensation. The compensation is that they receive the X chromosome from the father that contains W plus allele, that is for red eye. And because they become heterozygous, all females are to be expressing only red eye phenotype. But all of these males are to be expressing only white eye coloration because they are always to be receiving the X chromosome from the mother that has W allele. So in this way, we can be correlating with how we have the X chromosome inheritance in the male and female flies, showing a similar pattern to that of the inheritance of eye color gene, that is the W and W plus gene in Drosophila. And in that way, Morgan was able to provide an experimental evidence for the chromosomal theory of inheritance, where he states that the genes for the different traits are located on the chromosome, and that these chromosomes are passed from generation to generation, or passed from the parents to the progeny. Apart from the experimental evidence for the chromosomal theory of inheritance, Morgan and his colleagues conducted many dihybrid crosses in Drosophila to study the inheritance of two genes. Unlike Mendelian dihybrid cross, Morgan's crosses were between flies that differed in two genes which are located on the same chromosome. So he was trying to understand the inheritance of two genes which are located on the same chromosome. Let's take an example. One of such crosses was a cross between body color gene and eye color gene. So through his many genetic studies, Morgan realized that body color gene and eye color gene are both present on X chromosome. So, and he also knew that gray body was the normal body color in the flies, whereas one of the mutant body colors was a yellow body. So he denoted the yellow body with a small y letter and the gray body with y and a plus superscript, because gray is the wild type body color. He also knew that eye color can be the normal eye color, which is the red color, or it can be the mutant eye color, which was white eye color. So white eye was denoted with just a small w, and red eye was denoted with a small w with a plus superscript. Now with these knowledge, now let's try to look into the steps or the format of dihybrid cross conducted by Morgan. So for his body color and eye color dihybrid cross, what Morgan chose was, he chose female parents having both the mutant phenotype for body color and eye color. So the female parents were yellow bodied and white eyed, whereas he chose the male parents having both the wild type phenotype for body color and eye color. So his male parents were gray bodied and red eyed. Okay, now because we have to be proving that these genes or these alleles are to be carried on the chromosome, and because the chromosomes are to be inherited from generation to generation, so we are going to be denoting the chromosomes here. Okay, so for females, as we know, they have two X chromosomes. So the X chromosome is denoted by a straight line. All right, so here we have a horizontal line that is, so each line is denoting one of the X chromosomes, and we have two X chromosomes in females, so we have two of the red line here for the female parent. And because we know that male parents have one X and one Y chromosome, so these are different chromosomes. So X chromosome again is the same. We are going to be denoting it with a line, but Y chromosome, to have to differentiate it from the X chromosome, we are going to be having a hooked kind of a line. Okay, so this denotes the Y chromosome and this one is the X chromosome, and I'm using two different colors for the male and female. Alright, so now we have to also write down the genotype or the allele for yellow body and white eye for female parent, and gray body and red eye for male parent. And we know that these are all located on X chromosomes. So now yellow body is denoted with just small y, white eye is denoted with small w, and the female here is a homozygous female. So we have small y, small w, carried on the two of the X chromosomes. So both of the genes are located on the same chromosome. We have two genes on the same chromosome. Okay, now here in the male parent, they just have a single X chromosome. So on the X chromosome, we are going to be showing with the wild type body color and wild type eye color, which is Y plus and W plus. So we are going to be keeping the order of the letter consistent. So we have Y plus and W followed by W plus. Okay, carried on the X chromosome of the male parent. Then we can think about the segregation of gametes. Now, at the time of the segregation of the gamete during meiosis, we can be having females producing all of the similar type of gametes, because female here is not only they have the same sex chromosomes, but they also have the same allele. They are homozygous for yellow body and white eye. Okay, and because of which we can be expecting, even though we can be expecting 4 of such gametes, but all of the gametes are going to be just the same type. So I've just shown one single female gamete here on the left, and then on top of the Punnett square, I'm showing the 2 different types of male gametes. So the male gametes are always going to be of 2 different types, because here the chromosome, sex chromosome itself is different. These are heterozygotes. Okay, they release different kinds of gametes. So the one sperm would be having X chromosome, and that X chromosome carries Y plus and W plus on its different location, and then the other sperm would be the Y sperm, or this. Alright, green hooked structure. Now then we can be making the possible combinations for F1 generation. So all of the F1 females are going to be having this combination, XX combination, where one of the X's is having YW, the other X is having Y plus and W plus, and because of which all of the F1 females become heterozygous. Okay, they have Y plus Y, W plus W. Alright, whereas the males are going to be having X from the female and Y from the male. Okay, and on the X they are going to be having Y and W. And if you think about the phenotype, so we say that here yellow is gray is dominant over yellow, red is dominant over white. So the phenotype of F1 females are all going to be gray bodied and red-eyed, or you can say it's going to be wild type. Okay, simply you can be giving wild type as the phenotype. Then for the males, now the phenotype is going to be mutant phenotype, because on there only X chromosome, the body color is Y, which is for yellow, and the eye color is W, which is for white eye. So they are going to be showing mutant phenotype for both of the genes. Okay, so this is how we have reached up till the F1 generation. Now next, what Morgan did was he did F1 selfing. Now when such F1 parents were selfed, we then have to get the F2 generation. Now in order to predict with the possible genotypes of the F2 generation, we have to release the gametes from the male and female parent. So F1, since F1 male parents are going to be having 2 different sex chromosomes. One is the X chromosome, the other is Y chromosome. So we can be always expecting 2 different types of male gametes. A sperm containing X chromosome and another sperm containing Y chromosome. But when we look into the F1 female parent, since the female parent is a heterozygous female parent, it has Y plus Y, W plus W. It is heterozygous for both of these traits. So during the time of gamete formation, we can be expecting 4 different types of gametes in this case. Okay, now how we can be expecting these 4 different types of female X are, if the 2 X chromosomes, because the X chromosomes are both same, they are homologous chromosomes. If these homologous chromosomes segregate from each other without any kind of a change, okay, so if they simply segregate from each other, then we can be expecting, or we can be getting these 2 possible X. Okay, so we have a red colored X, just as it was in the F1 female parent, and the other egg is black colored X chromosome, which is also the same that got segregated from the red X, and hence we have 2 of the parental X chromosome simply segregating from each other and giving rise to these 2 possible female gametes. And at the same time, what we can be also expecting is, because these are both X chromosomes, because they are homologous chromosomes, so we can be expecting a very important event, or a very important kind of an exchange that would occur between the 2 X chromosomes. So red X chromosome can exchange its information with black X chromosome, And in the same way, black X chromosome can exchange its information with red X chromosome. And as a result of which, when we have such kind of an exchange, which is known as crossing over. So if we have a crossover in the 2X chromosomes during gamete formation, and this occurs during the prophase 1 stage of meiosis 1. So if crossover occurs between the 2X chromosomes of the female parent, then this would be resulting in 2 new combinations. And the new combination are a red having a segment of the black chromosome, likewise black X chromosome having a segment of the red X chromosome. And as a result of which, now we have W plus moving up with Y and W moving down with Y plus. So you have 2 of the other female gametes here, Y, W plus, Y plus, W. Okay, so in that way, if the female parents are heterozygous, then we can be expecting 4 different types of gametes. Now, with these gametes written down on the Punnett square, now we are to be combining with all the parental gametes, respective parental gametes, and forming with their combinations. So what we see here is, in this column, since the offspring, or since this fly is receiving X chromosome from both of the parents, so these flies are going to be becoming the female flies. And these flies, since they are receiving one X from the female parent and the other Y chromosome from the male parent, so they are going to be becoming all male flies. But if you look into the genotype and with the phenotype of the flies, what Morgan observed was he could be finding 4 different phenotypes in F2 generation. And these 4 different phenotypes were yellow-bodied and white-eyed male and female flies. Likewise, he also had grey-bodied and red-eyed male and female flies. He had yellow-bodied and red-eyed male and female flies as well. And he also had grey-bodied and white-eyed male and females. Now, if you observe the 4 phenotypes of the F2 generation, you will notice that 2 of the phenotypes are parental phenotypes, because we used yellow-bodied and white-eyed phenotype in the parental generation. So yellow-bodied and white-eyed is a parental phenotype. Likewise, we also had grey-bodied and red-eyed parrots. So we also got in the F2 generation grey-bodied and red-eyed. So these 2 are the parental combinations. We call them as parental combinations. So 2 of the phenotypes had parental combinations of phenotype, which is yellow-bodied and white-eyed, and grey-bodied and red-eyed. Either they were purely mutant for both of the body color and eye color, or they were purely wild type for both body color and eye color, as it was the case in the parrot. Now, the other 2 phenotypes, which are yellow-bodied, red-eyed, and grey-bodied, white-eyed, is a completely new phenotype. So we call these as non-parental combinations. Non-parental combinations. Okay, and another very surprising result of Morgan's dihybrid cross was, even though he was able to be obtaining with 4 different phenotypes in the F2 generation, these 4 different phenotypes did not have the Mendelian F2 ratio, phenotypic ratio. So Mendel always had 9:3:3:1 phenotypic ratio in the F2 generation. So we can, when we look into the phenotypes, we can also find 4 phenotypes very similar to that of Mendelian phenotype. But for Morgan, the kind of difference that he observed was these 4 phenotypes did not appear in 9:3:3:1 ratio. In fact, what he saw was majority of, most of the F2 flies had these parental combinations of phenotype. So most of the flies had yellow-bodied, white-eyed kind of the phenotype, or they had gray-bodied and red-eyed type of phenotype. And very limited number, very few of the flies in the F2 generation showed with these yellow-bodied, red-eyed combination or gray-bodied and white-eyed combination. So this is how he was able to see with the difference between Mendelian dihybrid ratio to that of his dihybrid cross result. Morgan also calculated with the frequency of parental combination as well as non-parental combination in the F2 generation. What he observed was parental combination appeared in 98.7%, whereas non-parental combination appeared in only 1.3%. To confirm with this result, Morgan and his colleagues conducted many similar dihybrid crosses with other linked genes. And in all of their experimental results, what they obtained was, when two genes are located on the same chromosome, these two genes always tend to be sticking together or going together into the gametes during meiosis in higher proportion, as opposed to the two genes showing independent assortment. So Morgan explained that the higher frequency of parental combination that is always seen in dihybrid crosses where the two genes are located on the same chromosome is because of a phenomenon known as linkage. So he said that the two genes which are located on the same chromosome always have a tendency to stay or remain together, which is a phenomenon called as linkage. And because of such tendency to stick together or go together into the gametes, it can be resulting always in higher proportion of parental combinations. Whereas the way he explained with the non-parental combinations also appearing in F2 generation was, he said that non-parental combinations can appear in the F2 generation because of recombination. So recombination he explained as a new combination that can also be seen in the two linked genes because of crossing over. So if crossing over occurs during prophase 1 of meiosis 1, then we can be expecting the two linked genes to be showing independent assortment. Okay, so the two linked genes can be forming new combinations of the alleles, and thereby it can be resulting in non-parental combinations. But the non-parental combinations are always to be appearing in lesser frequency as compared to the parental combination. So overall we are going to be always having recombination in lesser frequency and parental combination in higher frequency. Let me now summarize two of the major crosses, dihybrid crosses conducted by Morgan. One is a cross between body color gene and eye color gene that we discussed in detail, and the genes are denoted with Y and W letter. So in such crosses, what he observed was parental combinations which can be Y-W combination as well as Y plus and W plus combination appeared in 98.7% of F2 generation, whereas recombinations which had Y-W plus type of combinations and Y plus and W type of combinations also appeared in a very low frequency with 1.3%. Now, in his yet another dihybrid cross, where he made a cross between eye color gene and wing size gene, and the genes are denoted with W for eye color, as we know, and M is used for wing size gene. So, wing size gene is again located on X chromosome. Now, just to give you a brief idea about wing size in Drosophila, so the normal wing size is dominant to that of the mutant wing size. So here the mutant wing size is miniature wing size, and the letter that we assign for miniature is a small m because it is recessive, and the normal wing is denoted by m plus superscript. So when he made the cross between eye color gene and wing size gene, what he observed was he had parental combinations with W and M combination. W is for wide eye coloration, M is for miniature wing size. So wide-eyed miniatured wing type of combinations, as well as W plus M plus combination, which here both of the phenotypes are the normal phenotypes. So red-eyed and normal winged type of F2 flies. So such type of parental combinations appeared in 62.8% of the flies. Whereas 37.2% of the F2 flies showed recombinations. So the recombinations were W-M plus type of combination, where we have W is for white eye and M plus is for normal wing. And he also had recombinations that showed W plus and M combination, which is the red eye color and miniatured wing size. And that appeared in 37.2% of F2 generation. So from these two crosses, what we can be observing here is that the overall frequency of parental combination whenever the genes are located on the same chromosome always tend to be showing in higher proportion or higher frequency. So we have higher frequencies of parental combination always whenever the genes are linked or carried on the same chromosome. But we can also be expecting non-parental combinations or recombinations in linked genes. And such kind of recombinations are always to be appearing in lesser frequency. So this is one of the basic idea that we obtained from the work of Morgan on the genes which are located on same chromosomes. Based on all of his experimental results between flies that differed in two genes which are located on the same chromosome, Morgan finally concluded with two new concepts. He proposed linkage and recombination. Linkage refers to the tendency of the genes which are located on the same chromosome to be always inherited together in the gametes as well as in the subsequent generation. Recombination refers to the crossing over of two homologous chromosomes during meiosis, which results in the formation of new combinations of alleles in the gametes. Now, taking example from Morgan's experiment, we saw that recombination frequency between Y and W gene was 1.3%, whereas the recombination frequency between W and M gene was 37.2% in F2 generation. So from these experiments, we understand that body color, eye color and wing size gene are all located on X chromosome in Drosophila. Now the next question that you might wonder is, why do we have a very huge difference in the recombination frequency of W and M gene and Y and W gene? So why the recombination frequency is comparatively higher in W and M gene and why it is very less in Y and W gene? Morgan proposed that the recombination frequency between two genes which are located on the same chromosome depends on the distance between the two genes. So he proposed that if the two genes which are located on the same chromosome are very close to each other, if they are tightly linked genes, then such kind of the genes would be always showing greater linkage strength and lesser of recombination. Whereas, on the other hand, if the two genes which are located on the same chromosome are further apart from each other or they are at a greater distance, then these genes would be showing lesser linkage strength and higher recombination frequency. So, in this way, he had the recombination frequency which was very low between Y and W because Y and W genes are very close to each other. So these are tightly linked genes, whereas he had a comparatively much higher 37.2% of recombination frequency between W and M gene because W and M gene were further apart. They were loosely linked genes, and because of which linkage strength was much lower in these two genes and recombination frequency was much higher. So in a nutshell, we can say that the strength of linkage is inversely proportional to the distance between the genes, and the recombination frequency is directly proportional to the distance between two linked genes. Finally, let me discuss the contribution of Alfred Sturtevant in the year 1913. Sturtevant was one of the undergraduate students in Morgan's lab, and he was only 19 years old during the time. Interestingly, what is being said is that Sturtevant collected all of the experimental data from Morgan's lab, and he then worked out the recombination frequencies between gene pairs on the same chromosome, and thus he was able to construct the first genetic map in one night. He actually constructed the first genetic map of X chromosome in Drosophila. So genetic map refers to linear representation of gene order on a chromosome. Now what Sturtevant proposed was, since recombination frequency relates to the distance between genes on a chromosome, so recombination frequency can be used as a measure to determine the relative distance between gene pairs on a chromosome. Now he expressed relative distance in map units. So Sturtevant considered 1% recombination frequency as equivalent to 1 map unit. Map unit these days is also called as centimorgel. So 1% recombination frequency was considered equivalent to 1 map unit or 1 centimorgel. Now, by calculating the recombination frequencies of all of the gene pairs on the X chromosome, especially he constructed the map of 6 of the genes on the X chromosome. So by working out on these gene pairs, he was able to position, or he was able to arrange the sequence of these genes on the X chromosome. What Sturtevant noticed was the genes Y and W, or the genes Y and O, always showed the least recombination frequency. And such kind of the low recombination frequency is because they are very tightly linked with each other. So he placed Y and W or Y and O gene very close together on the X chromosome because they showed very less recombination frequency. And he said that whenever the genes are tightly linked with each other, then we would be expecting greater linkage and lesser of recombination frequency. In the same way, when he studied the recombination frequency between W and O genes, what he observed was that the recombination frequency between W and O gene was 0%. Which means there was no recombination at all between W and O genes. And he called such genes as completely linked genes. So W and O genes are showing complete linkage. That means to say they are always going to be staying together or remaining together, and hence we cannot be expecting any recombination to be taking place between W and O genes. So if the genes are completely linked with each other, if they are very tightly close to each other, linked to each other on the chromosome, then they are never going to be showing any recombination. So recombination frequency would be 0%. Whereas he also observed that the recombination frequency between Y and R genes was the maximum. So they showed the highest recombination frequency, and because of which he realized that the distance between Y and R is of the greatest distance. And then the distance between W and V, or the distance between W and M, or the distance between V and R, or the distance between M and R was greater as compared to the distance between Y and W, because they showed a higher percentage of recombination frequency as compared to Y and W. So whenever the genes are loosely linked with each other, or when the genes are located on different chromosomes. Such genes are considered as unlinked genes. So some of the genes are either they are located on different chromosomes, as we found in the case of Mendel's trait. So all of the 7 pairs of contrasting traits in pea plants that Mendel studied were located on different chromosomes, and because of which, since they were all unlinked genes, they always tend to be expressing with the 9:3:3:1 ratio of independent assortment. Likewise, if the genes are located very far apart from each other on the same chromosome, but they are very far apart from each other, or they are distantly placed on the same chromosome, then such genes can also be showing very high degree of recombination and very less of linkage. So Sturtevant actually suggested that if the genes are unlinked, if they are very far from each other, or if they are present on different chromosomes, then such genes are to be expected to show more than 50% of recombination frequency. Whereas if the genes are located not located on the same chromosome, but if the genes express recombination frequency between 0 to less than 50%, then such genes he suggested as incompletely linked genes. So genes can be incompletely linked, or genes can be completely linked on the same chromosome, or genes can also be having unlinked nature. That is to say, they are either very far apart from each other on the same chromosome, or they are located on 2 different chromosomes, and basing on which we can understand with different degrees or different percentage of recombination. So this is how Morgan and his student Sturtevant was able to propose with the concept of linkage, recombination, and finally he also worked out with the first genetic map of Drosophila on the X chromosome. So with this, I come to the end of today's lesson, and I will catch up with my next video lesson on sex determination.